Over here! I'm trying to tell you something! Something important!
Paper chromatography is a form of chromatography that uses filter paper as the solid phase and allows molecules to separate based on size or solubility in a solvent.
To start the chromatography, you put water in the bottom of the beaker, just so it covers the bottom. To start, we're using blue marker to put 2 dashes on the filter paper, and red marker to put 2 dashes on the other side of the filter paper. You put the filter paper in and wait for the water to go up the filter paper.
Who's going to win? Blue? Red? Molecules separate as they move up the paper. The distance the molecules travel depends on their size and solubility in the solvent.
Smallest, most soluble molecules travel the farthest.
A glass plate is spread with a thin layer of silica gel, which serves as the stationary phase. A sample is then added at one end, where a buffer creeps from the sample side to the opposite end, carrying soluble molecules along with it. Molecules are deposited based on their size and solubility in the buffer. If the molecules are colorless, they might have to be visualized with developer. A chromatographic column is a tube with a membrane or a frit near the base. Resin beads of a particular type are poured into the column. The resin beads settle onto the frit and form a matrix through which molecules can pass. When the sample is added to the top of the resin bead, gravity will pull it into the matrix. Depending on the resin selected, the molecules in the sample will either bind to the beads or pass through them. As the sample drips out, it is collected at regular intervals into vessels of manageable volumes called fractions.
How a column works. The column trial.
Alright, so the orange is the resin, and we are now going to pass our samples through the column. They're going to be collected as fractions. The sample is added to the resin at the top of the column, and gravity pulls the sample through the resin bead matrix depending on the resin. Molecules can be separated based on size, shape, or charge. Samples that pass through the column are collected in small volumes called fractions. Under the ideal circumstances, molecules that separate in the column are collected in different fractions.
These are the fractions we collected.
Gel filtration chromatography separates proteins based on size using size exclusion beads. Ion exchange chromatography separates molecules based on their overall charge at a given pH.
Okay, now let's talk about the affinity chromatography.
Affinity chromatography separates proteins based on shape or attraction to certain types of chromatography resin.
Oh my god, Divas! Like, did you totally know that chromatography can be used to assess certain amino acid compositions in a sample? Like, no way! It can also be used to determine the RF value, which is the distance the amino acid spots travel compared with the solvent front. Oh my god! Other chromatography, such as gel filtration, ion exchange, affinity, can be used in manufacturing. What? Chromatography can also be used to isolate proteins.